So Telly said there's no way I could finish this in 15 minutes, and I'm going to prove her wrong. Okay, so I'm going to talk a little bit about some projects that have been going on in my lab about the metabolic contributions to ALS. Here's Jean-Martin Charcot, the father of who established the diagnosis as the set of symptoms as a diagnosis 150 years ago. There's a lot of interest in a variety of different causes that are mediators or contributors to ALS, and I'm just going to focus a little bit on the metabolism angle. So if you just go to Medline and cross neurodegeneration with mitochondria, you find almost 8,000 papers. And in ALS, it's been widely described over decades that there are metabolic defects in ALS. These are some of them listed here, and mitochondrial defects are incredibly common, but they are they've been seen in almost every single model from one way or another. Mitochondria are affected. So just to keep it really simple, you have fuel, mitochondria turn it into ATP. ATP is the energy currency of cells. So we had done some work several years ago to look at mitochondrial function in the SOD model of ALS. And here I'm looking at oxygen consumption. It's the slope of the line when ADP is added. That's the measure of oxygen consumption. And in non-transgenic animals, we have a certain rate. And in animals that have the ALS phenotype, it's less. So that means that the mitochondria are not functioning as well. So we wanted to study this phenomena, and we needed to develop a model. See, metabolism is a very complicated subject because different cells have different metabolisms and talk to each other. And so we wanted to simplify the system, but engage the critical elements. So it's very, very important to understand that synaptic activity and intermediary metabolism are intimately related. In fact, more than 50% of the energy consumed by nerve cells is driven by the synaptic activity. So if you want to study the energy economy of neurons, you need to have a system that includes, in my opinion, synaptic activity. So it would be the equivalent of studying a car that was sitting in a showroom versus one that's driving down the street. So we designed a platform for looking at energy metabolism, and these are just pure neurons grown in tissue culture. And when you let them go for 2 weeks, they have many of the markers of neurons, as you can see on the Western blots, and also with imaging studies. They have all of the components of synapses. So these are synaptically connected active nerve cells, but there are no glia. Okay. So the way we explored this was to plate the neurons, let them go for 2 weeks, let them develop synaptic connections, and then infect them with a virus that expresses wild type or mutant forms of familial ALS genes. And then after 2 days, replace that media with a media that has heavy atom carbon labeled glucose, so that we can follow the metabolism, follow the utilization of glucose by nerve cells. And what you end up finding is, so I'm actually going to summarize about 5 years worth of work in 2 slides to say, A, there's a lot of information, and boy, there's really a lot of information. And now I'm going to just summarize it because I only have 6 more minutes. The critical thing is that the label in glycolytic intermediates in glycolysis is down, very much down. The amount of label that's in the TCA cycle is up, very much up. So what that means is that the nerve cells have rewired metabolism in the setting of familial ALS genes in neurons. Now, I happen to be incredibly fortunate in many, many ways. But for this project in particular, I was able to make a connection with a guy named Jerry Donnell. Jerry Donnell is the world's expert in metabolism, and he helped me to read through these data and try to understand things. And one of the things he said was, there's too many carbon skeletons in the TCA cycle that could be accounted for by what's going on in glycolysis. So maybe there's another source. And so what he suggested I do is to look at the utilization of amino acids. So here we basically did the exact same experiment, cortical neurons, synaptically connected, expressing familial ALS genes, but now I'm feeding heavy atom valine and isoleucine, which are branched chain amino acids. And the long and the short is that, in fact, these amino acids are being used as fuel. So under normal circumstances, fuel is being used by mitochondria to generate ATP, and it's overwhelmingly glucose. Whereas in the setting of mutant neurons, they're both using amino acids as well as glucose. So that turns out to be important, I think. So you could think of it this way, that the TCA cycle is just starving for carbon skeletons to oxidize. And their resolution of that conflict is to use amino acids. So I should mention, we also looked at the levels of ATP, ADP, AMP, the redox state. There were no differences. You could not distinguish neurons that were uninfected with viruses that expressed wild-type SOD or wild-type TDP43 or the mutants. So at this stage of the disease in the dish, the neurons have rewired their metabolism very successfully. So we're modeling an early aspect of the disease process. And the evidence that they've done a good job is that there's no change in the energy charge or redox state. Okay, I'm going to change the topic slightly. So we hypothesized that reduced glycolysis is starving the TCA cycle, and the TCA cycle is working overtime when acetylcholine enters. And if this is true, then maybe if we could stimulate the influx of glycolytic intermediates into the TCA cycle, it would be beneficial. So I happened to be reading the New England Journal one day, and I came across this article. It has to do with a type of hemolytic anemia. But this company, Agios, had made a pyruvate kinase agonist. So we took those pyruvate kinase agonists and applied them to our culture system. Now we have spinal cord neurons growing on astrocytes. We express FALS genes, familial ALS genes. And when you stimulate pyruvate kinase, you actually are neuroprotective. So do I have a pointer here? Hang on a sec. Oh, cool. I do. Okay, so pyruvate kinase is here. Okay, that's this step in the glycolytic pathway. And what I just gave you is some evidence that stimulating pyruvate kinase, which push more pyruvate into acetyl-CoA into the TCA cycle, is neuroprotective. So it turns out that the next step is catalyzed by pyruvate dehydrogenase complex. It's a complicated enzyme, but basically what it does is it is a decarboxylation and you generate acetyl-CoA. It's a huge, huge complex with 216 subunits, 9.5 megadaltons. I mean, this is a big guy. Okay, so it turns out that pyruvate dehydrogenase complex is inhibited by a kinase called the pyruvate dehydrogenase kinase. And it turns out that there's an antagonist of that kinase. It's called sodium phenylbutyrate. And what we found is that when you treat neurons that are expressing TDP43, mutant TDP43, you can kill them. And in the presence of sodium phenylbutyrate, you can suppress that effect. So sodium phenylbutyrate has many targets. In addition to the pyruvate dehydrogenase complex, it also affects another pathway. And in this pathway, branch chain amino acids such as isoleucine and valine first get their amino group is clipped off, you get a keto acid, and then the keto acid is decarboxylated. And it turns out that branch chain amino acids get turned into acetyl-CoA, And that's also taken care of by a branch chain keto acid dehydrogenase complex, which is inhibited by a kinase, and that kinase is also inhibited by sodium phenylbutyrate. So two different ways of getting more fuel into the TCA cycle are protective, and sodium phenylbutyrate and or the pyruvate kinase agonist are protective. So what we think might be happening is that by increasing carbon skeletons into the TCA cycle, that we are assisting in the rewiring of metabolism in a beneficial way. So maybe we're getting a little clarity. So Telly, I did it in less than 15 minutes. This is basically the conclusions. I just went over them. And here are all the people that I'm incredibly appreciative of who have done all the hard work. I want to, in particular, thank the people in my lab right now, as well as Jerry Donnell, who's a guru of intermediary metabolism. And if you have any questions, I can answer them later, because I think we're trying to keep on schedule.